Welcome. Today I'd like to present to you my research entitled A Comparison of the Degradation Rates of Polyethylene in a Novel Bio-Based Plastic. Each year, 8 million metric tons of polystyrene and polyethylene plastics contaminate our soil and water ecosystems as they are dumped into landfills or end up in bodies of water. Because these synthetic polymers are non-biodegradable, they ultimately create patches of microplastics in the oceans that biomagnify up the food chain to apex predators including humans. The health implications are massive. In order to combat this white pollution, consumers and companies are turning to biodegradable plastics to reduce the effects on the environment. However, the term biodegradable only indicates that the material breaks down into smaller pieces, yet still contributes to microplastic pollution and gives consumers a false sense of being environmentally conscious. Production of starch-based bioplastics are gaining popularity, and while they are truly biodegradable, the utilization of a food source may not be the best option for replacing the large plastic market due to the negative consequences of the fertilizer and land usage needed to increase corn production exponentially. To counter this issue, I set out to design a novel bioplastic that incorporated bamboo mainly because of its abundance and renewable qualities. It is also a source of tannins, seen here, a biodegradable anionic polymer that is gaining interest due to its practicality and suitability for mass production. After further research and experimentation, I chose to include chitosan, which is derived from chitin, when the degree of deacetylation is above 60% as seen here. Agar-agar was also chosen for its eco-friendly nature. Both of these substances react with tannins to produce a complex of strong chemical bonds. In this research, I compared my final product's biodegradability in both soil and water with that of a traditional petroleum-based film. I hypothesized that my formula would degrade much more quickly than the polyethylene film it was compared to, and have a comparable solubility with little water absorption. The experiment was done in two parts, the formulation and selection of the bioplastic, and then a biodegradation comparison of the new film to polyethylene. So to create the bioplastic, fresh bamboo culms were cut into small pieces and boiled at around 95 degrees Celsius in distilled water for approximately 200 hours until the solution darkened, indicating the release of tannins. To ensure the solution was the correct concentration, colorimetry was used to take a turbidity reading. A standard curve for the colorimetry had to be made, so 6 samples were created with varying amounts of hydrolyzable wine tannin, as I needed to know each of their concentrations. Once I had samples where I knew both the concentration and turbidity, I could use the trendline created by these values to find the concentration of my unknown where only their turbidity was known. After the tannin solution was the correct concentration, 0.3 grams of chitosan was mixed with 5 milliliters of acetic acid and then transferred to the tannin solution in order to dissolve the chitosan and bring the entire mixture to a pH of 5, which was the isoelectric point needed for the complex between the constituents to occur. 5 grams of agar-agar was added along with 0.1 milliliters of glycerin, and the solution was thoroughly agitated. After heating the mixture to approximately 95 degrees Celsius, the solution casting method was utilized by filtering the solution through a sieve with an aperture diameter of 2 millimeters and allowing it to solidify in silicone molds. 9 months of preliminary experimentation took place to synthesize the final bioplastic formula, with 60 various formulas tested before the final selection. This diagram shows the process I used to tweak the formulas once I had a basis. Overall, I found that glycerin, a plasticizer, was primarily responsible for the flexibility of the plastic. Agar-agar added stiffness and strength, and chitosan accentuated this strength, but was also responsible for some unwanted precipitation. The levels of acetic acid and tannins remained relatively constant as their concentrations seemed to be effective. The table shows the final 9 formulas examined and the results of the transparency, strength, and fatigue tests. These 3 were conducted to get a general overview of each formula's performance. So to determine strength, first, the samples were cut into dog bone shapes. An apparatus was devised to test the amount of weight a film sample could withstand before breaking. The scale on the right here shows the ranges of force in newtons that correspond to each point value. Next, UV spectrophotometry at 600 nanometers was used to determine percent transmittance, or transparency, of each film. Here's the grading scale that I used for this test. And finally, a fatigue test involves subjecting each film sample to continuous 90-degree bilateral flexes until breaking occurred, as shown in the diagram. And to the right is the grading scale for this test. So at the conclusion of the preliminary test, the points were summed, and whichever film had the most points would be the final formula. Formula 9, here in green, was selected despite the tie with sample 6, as strength was valued more so than transparency. Part 2 involved performing an in vitro biodegradation test in soil. Several months prior to experimentation, several plastic products were buried about 20 centimeters deep in outside soil to mimic the conditions of a landfill and lure out any plastic-consuming microorganisms. 20 borosilicate test tubes were filled with 8 grams of soil collected near the location of the pseudo-landfill. 10 were filled with a strip of the bioplastic and the other 10 filled with a polyethylene strip. A half a milliliter of distilled water was added to the surface to maintain the moisture content, with the same amount added halfway through the exposure period for the same reasoning. Trials were incubated at approximately 25 degrees Celsius for 168 hours. After the samples were removed and dried, Fourier Transform Infrared Spectroscopy, with the attenuated total refraction attachment, was performed to estimate the percent degradation of both samples by comparing the peak ratios of the spectra. Next, water absorption, in addition to solubility, was calculated. 15 10 mg samples of both the bioplastic and polyethylene film were submerged in distilled water for a period of either 1, 5, 15, 30, or 60 minutes. Samples were gently dried to remove any water on the surface and reweighed in an analytical balance. The remaining solution was then transferred to glass sample vials in order to determine turbidity and therefore solubility by comparing the turbidity to that of a solution containing one completely dissolved strip. FTIR was used once again to view any distortion and act as a secondary method of determining the percent solubility using an untreated sample as a comparison. 3 t-tests assuming unequal variances were performed between the biodegradation rates and water absorption and solubility percentages of the bio-based and polyethylene films. And here's a complete list of all materials. Analysis of the soil degradation FTIR spectra showed 22% degradation after 168 hours for the bioplastic, while only a 2.2% degradation for the polyethylene film. The t-test suggested that the difference was not significant with the p-value equaling 0.0635. However, assuming the biodegradation was constant, the biodegradation rate of the bio-based film is approximately 5,700 times faster than its polyethylene counterpart. The reasoning for this difference is mostly due to the unnatural bonding of polyethylene, seen in the FTIR spectra. While carbon-carbon bonds aren't unnatural, the long chains of them are, making it difficult for any microorganism to break it down. Seen around the 1050 mark, the bioplastic contains carbon-hydrogen bonds, and while they are considered stronger than carbon-carbon bonds, they are abundant in nature, making them no match for organisms. The difference in water absorption between the two films was significant throughout all times tested. The polyethylene film had a maximum absorbance of 20%, while the bioplastic's absorbance increased from 110% of its weight to 206% from the 1 minute to 30 minute marks until it began to plateau and eventually decrease. It is suspected that the decrease in absorbance was due to the dissolution of the sample between the 30 and 60 minute marks. The polyethylene film had a 0% solubility for all trials, but the solubility of the film reached its maximum at the 60-minute mark with 18.2% solubility. It may be inferred that this trend would continue to increase inversely with time. Further investigation suggested the presence of aliphatic amines, which are molecules that form hydrogen bonds in this scenario. A secondary peak in the first region of the spectra indicates that the suggested hydrogen bonding is present. Nitrogen and hydrogen, or oxygen and hydrogen bonds are polar. Using the principle that like dissolves like, it is understandable why the bio-based plastic is soluble. The FTIR spectra analysis of the bioplastic after exposure to water revealed that there was some distortion of the carbon bonds. The solubility values for the bioplastic obtained from the FTIR analysis differed by approximately 12.4%. The reasoning for this may be that some residual water was left on the sample strip after the testing concluded, leading to continuing dissolution. However, the general trend of solubility over time is the same for both methods. A cost analysis considering only the major constituents of each film estimated that the bio-based plastic would cost approximately $0.0016 more per cubic centimeter than traditional plastic due to the eco-friendly products, with an even smaller gap when considering recycling costs. To conclude, the novel bioplastic formula demonstrates potential after analyzing its effectiveness compared to petroleum-based film. Its biodegradation rate was impressive for the short exposure time, but its rapid absorption of water is a hindrance in its application. Additional tweaks may be made to the formula to improve its quality, such as altering the amount of glycerin. Further testing may include tensile strength testing coupled with film elongation testing. The tannin's antimicrobial and antifungal properties may be valuable in its use as a food rep applied as a spray-on or immersive solution. Its organic composition may also prove to have great promise in the medical industry as a bioabsorbable surgical implant, a method of implanted drug delivery, or other moldable medical device which might require post-surgical dissolution. Thank you for your time, and I hope you enjoyed this presentation.